So our last talk in this session is going to be by Dr. Stephanie Sarantopoulos, and she's on the heme malignancy and transplant faculty at UNC. She spends a lot of her time when she's not in the clinic working on issues of how do we deal with one of the major complications of transplantation, which is graft versus host disease. And Stephanie's been a leader in what the role of B cells is in that process, because traditionally we think of that as a T cell mediated problem, but in fact she's discovered that the B cells play an important role as well. So Stephanie?
I think the biggest reason people were relieved, and the organizers in particular, that I stood in for Dr. Bird was that my name is so much easier to spell than his. So I hope you've learned a bit about passive immune therapy from Dr. Richards, and then about the use of complex regimens in multiple myeloma that use immune modulatory agents like Revlimid. I want to talk about the only standard active immune therapy we have available for cancer patients, and that is donor or allogeneic stem cell transplantation. Patients who come to us have refractory hematologic malignancies, and we utilize allo transplant because it entails the transfer of a donor stem cell product that, while matched at HLA, is otherwise genetically disparate from the recipient. And it's the genetic disparity between donor and recipient that confers the immune response required for abrogation of remaining tumor cells. So it's the graft versus leukemia effect that's desirable as a therapeutic agent, but it's the unfortunate inextricable link with this graft versus leukemia effect to normal tissue destruction or graft versus host disease that makes this terribly toxic as a therapy, and one that requires further research. And I'm not going to tell you about acute graft versus host disease today, but I put it up here to tell you that it's quite distinct clinically, not just temporally, from the disease, chronic graft versus host disease. So chronic graft versus host disease occurs 7 to 12 months post-transplant, but clinically it looks for all the world like an autoimmune disease, like scleroderma or Sjögren's disease. And that makes it a clinically quite distinct entity. It also is the biggest problem our patients face who are otherwise cured of their hematologic malignancies. So this is a slide courtesy of Stephanie Lee and Mary Flowers at the Hutch in Seattle, showing you what I mean. So severe chronic graft versus host disease is the leading cause of non-relapse mortality in patients who've been otherwise cured of their cancer, and it remains to date a problem that is increasing in incidence and severity, not decreasing, despite our use of prophylactic agents like calcineurin inhibitors and such. And the reasons for this really are that we just simply don't understand the disease, and we have to do a lot more work on pathophysiology, and that's what I'm going to share with you today, is how we are doing hypothesis-driven science in the lab using human samples to try to understand the disease. And I use this slide not to, just to remind you, we can't also understate the morbidity that our patients experience. So, we cure our patients, but then they can't go to work. And so we really have a lot of impetus to try to develop targeted agents. What has been studied over many years, over 30 years of research, has resulted in some interesting and very important information. And I don't want to leave the T cells out of the discussion, because obviously T cells are very important in inciting the disease. And we know that from lots of murine studies, over 30 years of murine studies. But we also know when we T deplete our patients, they don't get chronic graft versus host disease. Unfortunately, when they don't get chronic graft versus host disease, they relapse their disease. So again, that inextricable link between GVH and GVL is important. Our work is focused on B cells, and I hope to convince you that what we're trying to do is separate that GVL from GVH by focusing on targeted agents for specific activated B cells. And the data for B cells has been around for a long time. So the folks in Seattle, Rainer Storb's group, first identified immune complexes in tissue of chronic graft versus host disease patients in 1978, and then everyone forgot about the B cell for a while until the rheumatologist helped us remember a few things. And it actually is interesting, I think, to know as a translational scientist that the murine studies are coming out now in 2012, and those studies actually followed the human experiment, if you will, the phase 1-2 trials using the available agent that targets B cells, rituximab. So in patients who had steroid refractory chronic graft-versus-host disease, when they received an anti-B cell agent, rituximab, there was clear clinical efficacy. And we see that it's a short-lived efficacy, but it definitely is proof of principle that compels us to further study the B cell in our patients. And what I thought about when I first started thinking about the problem around the time of completion of that rituximab, those two rituximab trials, one in Dana-Farber in Boston and one in Italy, the GITMO trial, was that there were alloantibodies and autoantibodies discovered in patients who had chronic graft-versus-host disease develop. And the alloantibodies are, were really the strongest reason why people used rituximab. But again, alloantibody responses may play a role in anti-tumor responses, and it would be hard to rationalize targeting B cells that might have anti-tumor effects. Autoantibodies, however, pointed to this idea that while alloantigens incite the process, it's the break in B cell tolerance resulting in autoreactivity that one could probably separate out and target if we knew what we were doing. And the question was, what are these antibodies doing? We don't exactly know that yet, I'll just tell you, but we do know they are there in tissues, and there's lots of reasons, as Dr. Richards told you a couple of them, as to how antibodies could certainly cause disease in tissues. We wondered, what are the antibodies being produced by, if I can get the slide up, and who is driving the B cell. And so when we thought about these autoreactive B cells, and I hope to show you some data from our lab about these particular cells, a prime candidate for further study was this molecule BAFF. So B cell activating factor of the TNF family member. TNF family is a very interesting molecule. It was discovered in 1998, and it's known to be pivotal for both the maintenance of B cell homeostasis and the development of autoimmunity. So we know if you overexpress this molecule in mice, you get what looks like a lupus phenotype. And if you knock it out, you don't get mature B cells developed at all. So we know it's a key factor. It's interesting because it binds to any of 3 B cell receptors on B cells and is primarily made by non-B cells. And it was a factor that we could measure in patients. So in this first analysis done at Dana-Farber, in 104 patients and some healthy controls, we did a very simple thing, which was just to measure soluble BAFF here on the y-axis in patients post-transplant and compare them to healthy individuals. And the first thing we noticed was that patients without chronic graft-versus-host disease had a significantly higher BAFF level in their plasma compared to patients with or healthy individuals. But importantly, patients with chronic graft-versus-host disease, both extensive and limited, had high BAFF levels, suggesting that this was a relevant molecule to further pursue. And when we looked in this patient cohort, and this has been confirmed in other patient cohorts, but by univariate analysis, BAFF was the only factor compared to other prognostic factors post-transplant for chronic graft-versus-host disease that was significant statistically. And logistic regression confirmed this factor to be also important. In fact, it was the only significant factor associated with chronic graft-versus-host disease in patients who were about 19 months post-transplant and had active disease at the time of sample draw. And to understand whether or not this molecule was really biologically relevant, we looked at the B cell, and we looked at the BAFF receptor on the B cell to see whether or not there was any evidence that BAFF was binding to the BAFF receptor. And fortunately for us, the antibody for BAFF receptor is not the greatest for flow cytometry. And what it is that when the receptor is occupied, BAFF receptor is no longer measured by flow cytometry, and I show you a representative example of this here. If you look by flow cytometry at whole blood of healthy individuals, patients after transplant without chronic graft-versus-host disease or those who had disease who are now healthy, there is high BAFF receptor expression on CD19 positive B cells as shown here. However, patients with active chronic graft-versus-host disease, the detection of BAFF receptor went down dramatically, suggesting this was occupied, and we have lots of other data to show that this is occupancy of the receptor. Suggesting that BAFF was doing something to B cells. And then an interesting one experiment that has prompted lots of fervor in our community, and I'll just show it to you for that reason, is we were able in this study to take patients who at 6 months post-transplantation had no evidence clinically of chronic graft-versus-host disease and look at their BAFF levels. And what we found was in this 43 patient cohort. Patients who went on, it's between 7 and 12 months to develop disease here, shown as yes chronic graft-versus-host disease. 77% of those patients went on to develop chronic graft-versus-host disease when they had a high BAFF level. On the other hand, patients with the low BAFF level, only 12% of them went on to get chronic graft-versus-host disease. To us, suggesting this is biologically relevant. To lots of other folks, suggesting that this could be used as a biomarker for chronic graft-versus-host disease. And I won't lie, we're looking to make sure we can understand whether we can use this for a biomarker as well. But we have a lot of things that I hope to convince you of that we have to do in terms of understanding the biology. But clearly, this is an ELISA. It's easy to measure. It'd be great if we could corroborate our clinical findings when we see our patients with the simple ELISA test. It'd be nice if we could predict who's going to get the disease, and certainly be great if we knew how to taper immune suppression better in our patients. So lots of people doing this, including us, and some work I won't show you in terms of the biomarker question here. And these are some of the subsequent studies that happened. So a group in Canada confirmed in children that chronic graft-versus-host disease patients had high BAFF levels at the time of diagnosis, and also had high BAFF levels that predicted their response to treatment of steroids. Response to treatment was also shown in another study by Whittle et al., showing that response to extracorporeal photopheresis correlated with decreased BAFF levels compared to those with high who did not respond as well. And then we can also look at polymorphisms in the BAFF gene in order to try to predict whether patients will get chronic graft-versus-host disease. And this was an interesting study out of Vanderbilt. And we, in collaboration with Dr. Richards, have looked further at some of these polymorphisms. And there's lots of work to be done there. But we're hopeful that not only does this confirm biologic relevance, but maybe we'll find a marker for disease. So I already warned you about the complex biology. But to put it in simple terms that I think are really interesting concepts to grasp here in terms of transplantation. BAFF has two roles I already hinted at with the mouse studies. One is a physiologic role, so you absolutely need BAFF to reconstitute your B cell compartment. There's also the pathologic role, so when it's overexpressed, you get autoreactive B cell clones surviving. And this molecule is quite complex in terms of its biochemistry. The trimer, the TNF normal trimer, is probably what confers the survival of normal B cells and B cell development. And there are other oligomer forms or other large oligomers of BAFF that likely confer pathologic signaling that we're trying to investigate. And the BAFF6 has been identified in mice, and it's something we're actively pursuing in our patients as well. The signaling and the activation of B cells can't be understood in isolation, however, so not to teach you all about B cell development, but this is a key concept slide. So if you think about B cell development in all of us, it's a dynamic, ongoing process. And the reason that none of us hopefully has an autoimmune disease is that your B cells emanate from the bone marrow every day and develop in the setting of exposure to antigen. If you didn't have what are called B cell tolerance mechanisms or elimination of autoreactive B cell clones, like you do, you would have 75% of your B cell pool in the bone marrow that would be autoreactive. But we have receptor editing and other immune mechanisms to avert this. The most important peripheral B cell tolerance mechanism is actually the limiting amount of this molecule BAFF. So if you have insufficient amounts of BAFF for potentially autoreactive clones, that's the important way to avert autoimmunity in the normal setting. And that's an important concept because when we look at our patients after transplant, we find a very interesting thing, which is exactly what has been found in these murine models of autoimmunity, and that is patients who develop chronic graft-versus-host disease have a very high BAFF level, but this is only important when the B cell pool, and the naive B cell pool in particular, is low. So you need a very high BAFF level per B cell in order to get the autoimmune phenotype, and we've shown this twice, once in the setting of the patients with active disease, and the second time in the setting of patients who get reinduction of B lymphopenia with rituximab. Those patients I told you about had clinical efficacy. Well, half of them went on and didn't do so well, and they in fact got worse, and that makes sense in the context of what I'm talking about here. When you don't have a naive B cell pool to sop up that extra BAFF, all those rare bad B cells are getting enough signal to survive and be activated. And naive B lymphopenia has been investigated further by our group, by Yuri Federu, who's our hematopathologist extraordinaire, and he looked at the bone marrows early post-transplant in patients and found that it's not just how much BAFF you have, it's really the production, the production capacity of the bone marrow to make new B cells that matters. So when he looked at day 30, bone marrows that had decreased precursor B cell numbers were the bone marrows of patients who went on to get chronic graft-versus-host disease, and the reverse was true. A robust B cell reconstitution was necessary to avert autoimmunity. And this is shown here. This is, again, a nice picture, I think, summarizes years of work. And this is if you have high BAFF levels, but you don't have a problem with your B cell compartment, you reconstitute a nice, normal, complex B cell pool, you're able to sop up excess BAFF, and the B cells that would be most reliant on BAFF for survival will be eliminated by deletional tolerance. On the other hand, if you have lots of BAFF around but not a lot of naive B cells, you end up with autoreactive B cells surviving and autoantibody production. And we're very keen on trying to understand the B cells coming out of the bone marrow and B cell production in the factory or the bone marrow. And I told you about Yuri's paper showing that precursor B cell production was vital for aversion of autoreactivity. So lots of B cells coming out of the bone marrow, good non-autoimmune phenotype, not a lot of B cells, that's not a good thing, and you end up with autoreactive B cells. So of course we are interested in that biomarker clinical application, but we're really also very interested in developing novel therapeutics, and that's the primary goal of my lab. We know that from what I just told you, that if we eliminated BAFF, we think we'd make headway with patients who have active debilitating chronic graft-versus-host disease. We're trying to get that agent. It's been FDA approved for lupus and rheumatoid arthritis. We're trying very hard to get it paid for our patients. Sounds like a common theme of this meeting, right? These agents are available, but we can't just get them for our patients. But we're hopeful and still trying. But meanwhile, we're going back to the factory of these autoantibodies and the B cell and trying to identify specific activated signatures of our B cells to see if those are going to be specifically targeted with small molecule inhibitors that are also available and that we think we can develop for our patients. So that's the next part of the talk I'll tell you about, which is the B cell signaling story and how we're using our tissue procurement effort at UNC in order to get at functional studies that we think will be important in identifying these targets. So BAFF is important for B cell signaling, and so is B cell receptor signaling. So our patients' B cells develop under constant exposure to allo and neo-autoantigens, and there's definitely a survival signal conferred by that. And then in conjunction, there's this BAFF signal. And this happens in normal B cells. We're trying to understand, are the B cells in our patients getting aberrant signals, and can we detect what those are? So our hypothesis was that BAFF was mechanistically linked to activation signals and survival signals in the B cells in our patients, and that we could identify them. And to do this, we have to have a large-scale effort tissue procurement protocol, and that's what we have at UNC. We're able to get every patient who comes into clinic who has a blood draw, give us an extra sample of blood, and we take those samples to the lab and right away phycol them and get purified B cells through immunomagnetic selection. And this is important for anybody who knows about how B cells behave. They don't like to live outside the organism, so we need fresh tissue trafficking, I call it all the time when we talk about our tissue procurement effort, to do these studies. And these are functional assays to show activation, and we can use antibody production, and we can use cellular protein content and cell size to measure and determine whether metabolic activation happens in these cells. And we can take these cells and do flow cytometric assays that will demonstrate whether the cells over time are surviving or dying, and do Western blots of whole cell lysates of these highly purified fresh B cells to determine whether signaling is happening. And our first clue that B cells were activated in the patients in vivo was to take B cells simply out of the body, highly purify a population of B cells we suspected were activated, and put them in culture, and measure over time IgG production. And what we found was very interesting, and that is patients who had chronic graft-versus-host disease had constitutive production of IgG, and that is you didn't have to stimulate them with anything, no anti-immune, no antigen, no nothing. They produced IgG automatically, suggesting that they had received an in vivo signal for this kind of activation. And I'll just tell you that there are lots of ways to phenotype B cells, and what we've tried very hard is to take what's in the literature and to understand that these are phenotypes, and we're trying to do analysis to find functional correlates of these phenotypes. And the first step was to find if there are uniquely circulating B cells that had at least the cell surface phenotype of an activated B cell, and indeed that's what we found. So patients who have active chronic graft-versus-host disease have cells that have high expression of IgD, and high expression of CD38, and also lots of the CD27 on it. And these are what are now deemed pre-germinal center B cells. And again, they uniquely circulate in our patients, are not found in post-transplant patients who have no autoimmune phenotype. They're not found in you and me either. And they're probably targetable cells. To get further information about these cells, we have to do large volume leukapheresis collections from our patients so that we can study these rare B cell populations, and that's just what we do. Again, so we can define functional correlates, and I'm going to show you that pre-germinal center B cell again here under the microscope. A picture's worth 1,000 words. That B cell compared to what is the most recent bone marrow emigrant, the transitional cell, is clearly different when you look. These B cells are large, they're surviving, and they look activated. And indeed, when you look at the total B cell population, lucky for us, you can take B cells, isolate them just by CD19 bead isolation, and put them in culture over time, and then measure apoptosis by an Xn5 staining. And what you find is that survival is improved, or apoptotic death is decreased in patients with active chronic graft-versus-host disease compared to other B cells from patients post-transplantation. And that suggests, again, a survival signal in vivo of these B cells. So the functional correlates happening here. Looking then further at metabolic activation of these cells, we did a simple thing, which was how much protein is in each of the B cells that we find in our patients with chronic graft-versus-host disease, and that's shown here. In a group of 17 patients, 9 patients with highly purified B cells had more protein per B cell compared to patients without chronic graft-versus-host disease, again, suggesting metabolic activation of these cells. And the size of these cells is increased as another marker of increased metabolic activation. So if you look by forward side scatter, by flow cytometry, the size of a B cell from a chronic GVHD patient is increased, and that held true for the cohort of patients examined. And signaling then was the next task, looking by Western blot, and we looked to the literature to find out what was being done in mice. None of this stuff, by the way, has been done in rheumatology either in humans, because they don't have the sort of capacity we do. We know when our patients are going to develop disease. We have the capacity to get the tissue. We know how to examine it. So I think our results will have some broad-reaching potential in that field as well. But what we did know was that in the mouse literature, there's clear signaling happening because of BAFF receptor tweaking with the soluble BAFF, and that is through activation of AKT and also activation of ERK, with the downstream consequence of decrease in isoforms of this pro-apoptotic molecule BIM. And that is just to show you that when we took our highly purified B cells and ran them out on Western blot and blotted with an anti-phospho-AKT to show AKT activation, the B cells from the chronic GVHD patients had activation through this signaling molecule. And the B cells had activation through phospho-ERK, or they were demonstrating phospho-ERK, where the chronic GVHD or the people who didn't have chronic graft-versus-host disease did not have signaling through ERK. And that held true for the whole group. And what we link, the way we were able to link mechanistically the BAFF signal to the effect we're seeing was by looking downstream at pro-apoptotic BIM. So if we found decreased BIM, we were going to be excited to say that this is likely BAFF-specific signaling, and indeed that's what we found. So B cell lysates run out on a gel and probed for BIM, demonstrated here, just shown as a representative example. Patients with chronic graft-versus-host disease were missing 2 of the isoforms of BIM compared to patients without disease. And again, that held true for 17 total samples post-transplant that we were able to examine in the lab. So I think using samples from patients and freshly isolated B cells in this case, and sort of trafficking that is required from the clinic to our lab, we've been able to identify signaling pathways that we think are relevant potential therapeutic targets for patients with chronic graft-versus-host disease, and specifically these would be activation of ERK and AKT, and I think I've shown that decreased apoptosis is the functional consequence of those activation signaling pathways, and metabolic activation as well, so probably through activation of AKT, increased protein content and size being the two measures that we used. And of course, as a person who sees patients with this devastating consequence of transplant, and our major goal is to try to develop existing agents that we have to target signaling pathway, and that's what we're actively doing now. So I hope I've convinced you that hypothesis-driven science using patient samples is possible, not only possible, but hopefully highly relevant. And I hope that we do develop preemptive therapies for our patients so that they don't get this devastating consequence. And thanks to everybody who took part in this research. And I have to make a big special plug to, one, the taxpayers, especially the taxpayers in North Carolina, because they pay for the University Cancer Research Fund that brought people like me here, and also continue to through the efforts and interest of Dr. Earp and our tissue procurement endeavor here at UNC, have made this research possible. So without the interest, the funds, and the infrastructure, we would be nowhere. And of course, the people in the lab are vital to getting this research accomplished. Thanks very much for your attention.
Thanks, Stephanie. We do have a few minutes for questions, so I think that probably 5 minutes or so. So if people have anything that they want to ask any of our three speakers, they're raise their hands. Let me just ask Stephanie one thing. The GVH picture is obviously dynamic, and it's, the timing of that is critical. But if you were to guess in terms of how normal B cell recovery occurs and trying to balance, having enough BAFF around to actually develop your B cell repertoire versus treating, would you treat GVH or would you prevent GVH, or what do you think would be the right time to give any kind of an anti-BAFF compound?
If you talk specifically about anti-BAFF, I think that's a tough question. We might actually be fortunate we didn't have the agent available, because it might have been used and then not successful because of that very question. So it's a very important one. I think that's why we're moving towards trying to develop other agents. I'm out of juice. Can you hear me? Okay.
Yeah, no,
so I think we don't know the answer in mice when you, and actually in the lupus patients, when you give an anti-BAFF agent, it's similar to what happens with rituximab. It's an all or nothing thing. It's an antibody. All the B cells are gone, and then once you stop giving the agent and the B cell reconstitution recurs, you get autoreactive B cell clones arising, and those will probably be divided again into those who get disease and those who don't. And so the answer to that is really those who don't have a good bone marrow. And so we have to do two things. One is give the anti-BAFF agent and then try to promote B cell reconstitution, naive B cell reconstitution. So the kinetics are probably not answerable, but I think the concepts are important to get at that, and we'd have to probably give it early, would be my guess, but with something that promotes B cell reconstitution at the same time.